Hello everyone, welcome you in my YouTube channel. I am Dr. Mohammad Sirajul Faizi, Assistant Professor, Department of Chemistry, LS College, Muzaffarpur. Today in this video, I am going to explain about proteins. What is the functions? You know the proteins, molecular tools of life, structural, cell shape, connective tissue, catalysis, enzyme, metabolic regulation, regulation of cellular metabolism, transport move substances back and forth across cell membrane, defense antibodies in immune response. General types based on 3D structure and functions role: fibrous, structural, membrane, several poles associated with the cell, globular transportations. So, produce from only 20 amino acids. 100 amino acid can produce 10 to the power 100 potential sequence. Subset structure and function is result of selection pressure. Structure feature facilitations, folding, presence of binding site, balance of structural flexibility and rigidity, appropriate surface structure, vulnerability to degradations, distinguished based on number and sequence of amino acids. Polypeptides, molecular weight 1000 to millions daltons. Peptide, lower molecular weight, less than 50 amino acid. Protein greater than 50 amino acid, one or more polypeptide chain. Amino acids, amino group and a carboxyl group bonded to alpha carbon. Amino group attached to the carbon adjacent to the carboxyl group. Side chain are bound to alpha carbon. R identifies amino acid. Look at this. Here, side chain R. This is alpha carbon. This is amino group. Here is carboxyl groups. And here is hydrogen. Amphoteric behaves as acid or base. pH 7 carboxyl group conjugate base form, amino group conjugate acid form. Zwitterion have both positive and negative charges. Non-standard amino acids chemically modified after incorporations occur in living organism but are not in proteins. All protein derived amino acid have at least one stereocenter, alpha carbon. Superimposable mirror image, a chiral glycine leg center of symmetry. Hydrogen as R group. Chiral stereoisomers are not superimposable. 4 groups bonded to alpha carbon. Side chain carbon designated with Greek symbols starting at alpha carbon, beta, gamma, delta, epsilon. So, what is the meaning of enantiomers? Molecules are mirror images of one another. Optical isomers, not superimposable and rotate plane polarized light in opposite directions. L-alanine amino acid on L, R-alanine amino acid on R, mostly L amino acid found in proteins. So name an abbreviation of standard amino acids. These are amino acids. 3-letter abbreviations, 1-letter abbreviations. Group A non-polar side chain: L-alanine, leucine, isoleucine, proline, phenylalanine, tryptophan, methionine. L-val-leu-IL-pro contains aliphatic hydrocarbons group. Pro has cyclic structure. Phi hydrocarbons aromatic ring, TRP indoles ring side chain aromatic, methionine sulfur atom in side chain, cysteine side chain contains thiol groups. So look at here: leucine, isoleucine, valine. Group B, neutral polar side chains easily interact with water through hydrogen bonding. These are side chain is polypolar hydroxyl group. Tyrosine hydroxyl group bonded to aromatic hydrocarbon group. Glutamine and ASN contains amide bond in side chain. So serine, threonine, tyrosine, asparagine, glutamine, they have different group here: OH, 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 amide, amide. Now group C, acidic side chains, both have carboxyl group in side chain, can lose a proton forming a carboxylate ion negatively charged at neutral pH. Look at here. Apart from this is extra. Group D, basic acid or basic side chain. Side chain are positively charged at pH 7. Arginine side chain is guanidino group. This guanidino. Histidine side chain is imidazole group. And lysine side chain is ammonia group is attached to an aliphatic hydrocarbons chain. So biologically active amino acids. Some amino acids or derivatives can act as chemical messenger. Neurotransmitters, glycine, glutamate, gamma aminobutyric acid, GABA, serotonin, melatonin. Hormones, thyroxine, signal molecule produced in a one cell, regulate functions of other cell. These are some derivatives of amino acids. Act as precursor for other molecules. Nitrogenous base component of nucleotides and nucleic acid, example heme and chlorophyll. Metabolic intermediate, arginine, ornithine and citrulline in urea cycle. This is citrulline, ornithine. Modified amino acid in protein. Derivatives of amino acid formed after protein synthesis. Serine, threonine and tyrosine can be phosphorylated. Gamma carboxyglutamate, prothrombin, collagen, 4-hydroxyproline and 5-hydroxylysine. Structural protein most abundant protein in connective tissue. Modified amino acid residue found in polypeptides. So titration of amino acids. Amino acid without charges groups on side chain exist in neutral solution as deuterion with no net charge. Cationic form, anionic form, and neutral form. So these are the titrations gram. This is simple amino acid, two ionizable groups, loses two proton in a stepwise fashion. Upon titration with sodium hydroxide, isoelectric point is reached with deprotonation of the carboxyl group. So I is equal to pK1 plus pK2 divided by 2. So pK values for the ionizing group of amino acid, these are amino acid, and these are the value of pK1 and pK2. Now you can calculate. Now glutamic acid has a carboxyl side chain group plus 1 charge at low pH. Isoelectric point between those of alpha carboxyl group portions, proton and R groups carboxyl portions. Proton. More base is added, it loses protons to final net charge of 2. You see, this is graph of pH versus adding OH minus. So friends, hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.